শোনা যা দিয়ে দেখা যাচ্ছে না ক্যামেরা
I think we are live now.
Alpana, are we online?
Yes.
Yes. Yeah. Thank you. Thank you. Yeah.
Just give us probably one more minute and then we'll start.
Sure.
Satnuji, we are good to go. Good morning, everyone. Welcome to our next round of science seminar in the Science Setu series. So today is World Cancer Day, and which is celebrated internationally to raise awareness of cancer and to encourage its prevention, detection, and treatment. So this seminar today is organized to create awareness about cervical cancer and oral cancer. You are more than welcome to forward the link to your family and friends who would be interested to join today's seminar. Along with World Cancer Day, this seminar series is also celebrated as a part of Azadi Ka Amrit Mahotsav, which is an initiative of the Government of India to celebrate and commemorate 75 years of Indian independence, its history and achievements. So these seminars, the seminar series the events as a part of Azadi Ka Amrit Mahotsav started from March last year and will continue on till August 2020. Now, for today's talk, we have two speakers. So our first speaker today is Dr. Ankita Chatterjee. She is a former PhD student from NIBMG and is presently working as experimental lab manager of the SIMEC project, the Systems Medicine Cluster project at NIBMG. Currently, she's working in oral cancer. So over to you, Ankita, and who better than you to talk about and create that awareness.
Thank you, Alpana. I am starting to share my screen. Is my screen visible?
Yes.
Okay. Okay, good morning, everyone. I would like to start my talk today on common cancers in India, and my talk will focus on early detection and prevention. So before I start, I would like to thank the Science Setu lecture platform for helping us to organize this lecture series, which will contain two talks. One will be given by me on oral cancer and followed by my junior and colleague on cervical cancer. And I would also like to thank all the audiences who have taken out some time from their busy day to join us in this lecture series. I hope that during the next 20 to 25 minutes, I will be able to give some insights on the prevalence of oral cancer across globe and also in India, and some early detection methods and preventive measures that can be taken easily. Today we are celebrating World Cancer Day. It's not exactly celebrating, but we every year we follow this day to try to generate awareness among the common people. And with this, I will start my presentation. So, cancer in the oral cavity. What is cancer? Cancer is uncontrolled growth of cells. So we know in our body there are multiple tissues, and these tissues take the shape and features and perform particular functions due to an organized functional role of the cells. And these cells grow and divide, but in an organized fashion. If it's uncontrolled, it will lead to cancer. So if we focus on the left figure, this is a figure of our oral cavity. It contains multiple parts. We know lip, teeth, gum or gingiva, tongue, hard palate, soft palate, etc. So this is a abstract picture of a normal anatomy of oral cavity. So what happens in cancer is this. So I have put three pictures of patients. I have taken most of the pictures used in this talk from internet. There are wide varieties of resources. So the first picture is of tongue cancer, as you can see from here, the gingiva-buccal cancer in the gum, and the lip cancer. And oral cancer does not only affect these three locations, but it can affect multiple locations within the oral cavity. It includes lips, cheeks, sinuses, tongue, base of the mouth, and it can extend up to oropharyngeal region. So how common are cancers in oral cavity? If we take the data from Globocan, it's a website which maintains a database of the prevalence of different cancers across the world, then we can see that there is a huge spread of oral cancer across the world, specifically in the Asian countries. So here I have put three figures with three different names. One is incidence, one is mortality, and the other one is 5-year prevalence. So incidence is like how many number of patients are detected in a particular time period, which is generally taken as maybe a year. So as you can see, Asia is showing the highest incidence rate compared to the other continents. Coming to the mortality, mortality is how much percentage of patients die within a particular time period, which is usually taken as a year. Again, Asian countries are showing, in the Asian countries, patients with oral cancer are showing highest mortality. And the third one is the 5-year prevalence. 5-year prevalence is if we take a span of 5 years, how much is oral cancer present across the world? Again, the Asian countries are showing the highest percentage. It's a large section of this purple that's showing the highest prevalence of oral cancer across world in the Asian countries. So what is the scenario in India? So India contains the largest number of oral cancer cases. It contains 1/3 of the total burden of oral cancer globally. And around 77,000 new cases and 52,000 deaths are reported annually in India due to oral cancer. So oral cancer is one of the most prevalent cancer type in India. who gets affected the most? So according to the Cancer Statistics 2020 report from Cancer Registry Program India, 1 in 147 males is affected in the tongue cancer, gets affected in tongue cancer compared to 1 in 401 females. So as you can see that the males are showing more, males are affected more with oral cancer compared to the females, and roughly we can take the ratio as 2. So whatever the prevalence of females with oral cancer, the males are the double. In mouth, 1 in 103 males gets affected, and in females 1 in 241, and likewise. The other thing is, if we take the age standardized incidence rate, if we standardize the age group across different parts of the Asia and Europe and world, how much incidence is seen in the males and the females. And as you can see, the purple ones are the female and the blue ones are the males, and the males are showing higher or taller bars, which is clearly showing that males have a higher incidence of oral cancer compared to the females. So it is really a potent health problem in India. So if we look at the spread shows that most of the oral cancer patients are detected at late age, above say 60 years, and they are mostly affected, males are mostly get affected, and there are certain risk factors which we will come one by one. And these risk factors, generally we can say that tobacco intake is the highest one. And this picture shows the prevalence of oral cancer across different parts of India, the north, northeast, central, west, south, and east. What are the risk factors? There are various risk factors detected for oral cancer. The first one is to point out is the tobacco intake. It can be either through smoking, like smoking through cigarette, bidis, or through chewing, like chewing of gutka. Along with that, the other risk factors include alcohol intake, chewing pan or betel quid, and also a very small percentage can be affected by viral infection, mainly the HPV. And as I have already mentioned, with increased age, there is a high risk of developing oral cancer. If we try to put the percentage of these risk factors determining the oral cancer, we will see that 49% of the oral cancer patients are detected due to the cause of tobacco intake, 10% due to alcohol intake, and 27% includes other risk factors. Along with these risk factors, there are genetic factors that can affect the occurrence of oral cancer in an individual. And in our institution, we try to find out the genetic variants or genetic changes that can cause the development of oral cancer. So a brief recap on how does our genome work. We know that our body is made up of cells, and each cell is made up of DNA, which lies in a condensed form called chromosome. And these DNAs are tightly packed, but essentially they are stretch of ATGC base pairs, which form the DNA. As cell divides, this DNA gets duplicated, and from a mother cell, daughter cells get generated. So synthesis of another strand of DNA from a DNA molecule, we call it as replication. DNA transcribes to RNA, and from RNA, it translates to protein. Now, proteins are the building blocks that carries out the downstream functions, and It determines the feature of a cell, it determines the function of a cell, etc. And this section, replication, transcription, and translation, as you all know, it belongs to the central dogma. So again, a brief through the central dogma. This is a double-stranded DNA. DNA, when it gets transcribed to RNA, RNAs are single-stranded nucleotide stretches, which when translates produce protein. Proteins are made up of amino acids. This is also important to remember that DNA from one individual can be different from the DNA of other individual, and the positions where the sequences are different, we call it as the variant position. With this basic knowledge, we find out that what are the genetic changes that can take place within our genome. We can classify the genetic changes as point mutations or chromosomal changes. Point mutation is when there is a change of a single DNA nucleotide. Chromosomal change is when there is a change or aberration in a chunk of the chromosome. A little more on the point mutation. So I am spending little time on establishing this fact because this will help us to understand what genomics can do in identification of causal variants or mutations in cancer. So what point mutations? Point mutations are different types. So at the DNA level, if we see a sequence as TTC, it gets translated ultimately to an amino acid called lysine. But if this particular TTC sequence gets changed, it can produce downstream numerous downstream factors. It can change the protein amino acids coded. So it's like if there is the C is changed to T, it will still produce a lysine. But if this T is changed to A, it will produce a stop codon in the mRNA so that no protein or amino acids are synthesized. So what this slide is trying to explain is the change in the DNA nucleotide sequence can affect the downstream protein structure, protein function, and change in the protein function will ultimately cause change in the expression, in the behavior of the cells, in the features of the cells. So we know the story of cell division, and it is actually a story of the accelerators and the brakes. So we know a cell, it first replicates its DNA and it divides, and from one cell it generates two other cells. These cycle, we call cell cycle, is tightly regulated by different checkpoint molecules. These molecules are again encoded by genetic segments or checkpoint genes. So essentially 4 major checkpoints are identified in the cell cycle. One can identify whether there is proper DNA has been synthesized. One can identify whether there is a damage in the synthesized DNA, and so on. So if we go I will not bore you with all the names of genes and details, but if we go a little bit into what are these genes that code for checkpoint proteins. So cyclin and CDK complex, these are the major players that regulate the checkpoints in the cell cycle. And one of the important function of this checkpoint molecule is to see that the mother DNA is correctly passed on to the daughter DNA, and a healthy mother cell divides into 2 healthy daughter cells. If there is an error, the checkpoint molecules try to identify that, and this will instruct the cell to stop division and die. We call the process as apoptosis. So apoptosis is a process of programmed cell death where the checkpoint molecule signals that there is something wrong in the cell division or cell cycle process, or you have something is wrong in you, you have to die. This is the normal cycle in which our body generates healthy cells, and ultimately each cell can perform different function in our body. But if there is a failure in this checking system, a normal cell can lead to a generation of abnormal cells, and they can proliferate highly, producing a tumor or cancer. So genetic alterations and cancer is a very, very intertwined phenomena, because this abnormal cell growth in cancer is tightly regulated by the genetic alterations. So sometimes what happens, and it arises through a series of mutation. It's not that only a single mutation can give the property of a normal cell to proliferate abnormally. Accumulation of multiple mutations can lead to the malignant cell and tumor growth formation. Now, two sets of genes play very important role in this malignant transformation of cells. One we call as tumor suppressor, and the other we call as the oncogene. So tumor suppressors and oncogene, their function balances the cell division cycle in a proper fashion. If there is a change or a mutation in these genes, generally loss of function mutation in the tumor suppressor and gain of function mutation in the oncogene, leads to cancer formation. So what are oncogenes? Oncogenes are like accelerators. They encourages the cell to proliferate, and if there is a gain of function mutation in an oncogene, it will do, it will give the oncogene additional capability to instruct the cell to divide more. And if we see this wall as a terminal point from where the cancer is developing, then activating mutation in oncogene can lead to the crash or cancer development. So there are genetic studies have identified some known oncogenes in cancer like RAS, EGFR, MEK, TGF-alpha. Again, what are the tumor suppressor genes? They are like the brakes. So they try to control or slow down the cell growth. Now, loss of function mutation in the tumor suppressor genes can lead to, again, crash or cancer development. But it's important to note that we carries two pairs of chromosomes, and if one brake gets defected, the other can still work. But if both of them get defected, then there will be complete crash. The most famous tumor suppressor gene is TP53. This gene is also found to be changed in a large number of oral cancer patients. So what is the involvement of NIBMG in the study of genetics? So in 2010, ICGC International Cancer Genome Consortium, along with Department of Biotechnology, Ministry of Science and Technology, set up a project to identify genetics variants associated with the development of oral cancer in India. And there are two collaborating teams, the Tata Memorial Center, ACTREC Mumbai, and National Institute of Biomedical Genomics. So at the Tata Memorial Center, they mainly perform the patient recruitment and biobanking and clinical and histopathological assessment of the cancer. While in our institution, in NIBMG, we perform the genomic analysis and try to identify the genetic variants or somatic mutations that can cause oral cancer development. So from the ICGC study, the study identified 10 genes, a set of 10 genes that were found to be significantly mutated in the oral cancer patients. So as you can see, TP53, which is a tumor suppressor gene, is highly mutated among the patients, followed by some other genes. And if you can see, this top 7 genes are mutated in 10%, at least 10% of the patients. So what this genetic study helped is to identify the mutations in genes that are mostly found in the oral cancer patients. And it can also help to identify in the understanding of the disease, and it can also help to identify groups of patients with different mutation profiles. Tumor suppressor genes commonly drive oral cancer. This was the major finding from the study done by the ICGC consortium. Now, from my talk, I think this picture will summarize. This picture is the first picture to summarize the hallmarks of cancer, where each of the points which I have mentioned are shown to be features associated with cancer development. But as the research progresses, more and more signals comes out, and genetic mutations or change in the nucleotide sequence in each of these factors are coming up one by one to have caused development of cancer. So coming back to the treatment regime of oral cancer, it is mostly a combination of radiation and surgery, and sometimes it's chemoradiation, where radiation is supplemented with chemotherapy. At stage 1, it's mostly single treatment procedure where it's radiation or surgery. But for stage 3 and stage 4, generally doctors go for a combination of treatment. However, there are some patients who recur, and even after surgery and radiation, and it's important to know what could be the reason behind these patients who have recurred after the treatment. So I hope some of you have read this book. If you have not, it's a very good start to know about cancer, know about researches done on cancer over the decades. And as the author stated, that the greatest need we have today in human cancer problem, except for a universal cure, is a method of detecting the presence of cancer before there are any clinical signs or symptoms. So early detection and prevention are the two most important factors that can help us to win over cancer. So key to oral cancer management will include early detection and prevention. In India, 70% of patients arrive at the clinic with advanced stage. And since it's in the late stage, the chances of cure are very low. The 5-year survival means a patient after its detection die within the 5 year, and the 5-year survival rate is only 20%. So what could be the preventive measures we can take? It's very important to stop intake of tobacco in any form. To avoid excessive alcohol consumption, eat a balanced diet, visit a dentist regularly, and lead a healthy life. Another thing is cell screening, which is very important for individuals who do not have to go to the clinic but can do regularly to identify any early signs of oral cancer. So these are the locations to check every day when you brush. And the early symptoms include a sore on the lip in the mouth that doesn't heal, constant pain in mouth, a lump or thickening in the lips, a white or red patch in gums, trouble in chewing or swallowing, trouble moving the jaw, numbness of tongue, swelling or pain in the jaw, etc. So I have summarized these early symptoms because this can help each and every one of us to regularly check our oral health and detect and go to the clinic if necessary. Finally, the theme of World Cancer Day 2022 is Close the Care Gap. The theme comprises of a 3-year policy where the first thing is to realize the problem of why there is a gap in the late diagnosis, why some people gets very difficulty during its treatment, and how can these kinds of problems be tackled, and finally, what are the challenges? So with this, I would like to thank all of you for your attention, and I wish everyone a healthy life. Thank you.
For all the attendees, you can type in if you have any questions in the chat box, and we will address them now and also after the end of the second presentation. So right now we don't have any questions, but we can maybe wait for a few minutes, for 1 or 2 minutes, and then we'll move on to our next presentation.
I think, Kalpana, we can go ahead with the next presentation, and at the end we can answer some questions if there are any. Thank you all for your attention.
Thank you, Ankita. Now, our next speaker today is Ms. Abarna Sinha. She is currently doing her PhD at NIBMG on cervical cancer. So who better than her to explain what it is and how can we prevent it? So over to you, Abarna.
Hi, am I visible?
Yes.
Okay, so I'm sharing the slides. Is it visible now? Okay. Hello, everyone. Good afternoon. I am Abarna Sinha, a senior research fellow working in National Institute of Biomedical Genomics. In earlier talk, Ankita has mentioned about genomes and cancer. So I guess you have got a brief idea about it. So you all have learned or heard about a tale of two cities. Today I am going to narrate to you all about a tale of two genomes. I would like to discuss one of the most common cancer among females, that is cervical cancer. The CA stands for cancer and CX for cervix. Now, where is this place located? This is a female reproductive system, and this portion over here is known as cervix. If any kind of cancer occurs in this region, it is known as cervical cancer. Why is it most common? Because statistics have shown that the incidence rate of cervical cancer is twice as high in the developing world than that of the developed countries, and mortality rate is also very high. It is 3 times as high in the developing countries than that of the developed countries. So you can see that it is very much prevalent in the developing countries. And India, being a developing country, you can see, why isn't my slide changing? Right. So India being a developing country, you can see over here the state-wise distribution that cervical cancer incidence rate is very high. So darker the color of the states, like Uttar Pradesh, Bihar, West Bengal, Maharashtra, all of these have high incidence rate of cervical cancer. But on the other hand, northeastern India, Jammu and Kashmir have lesser incidence rates. It is mainly seen to be occurring among Indian women belonging to a lower socioeconomic strata. Annual number of cervical cancer cases and deaths related to it is very high. It is around 62.42%. World Health Organization last year only have pledged to completely eliminate cervical cancer by 2030 so that there won't be any more cervical cancer cases. So you can see that it is pretty high right now, and within 10 years time only they have taken a pledge to completely eliminate it. And let me tell you that it is completely possible. It is a preventable cancer, and I will tell you how it is preventable. The main causal agent for cervical cancer is human papilloma virus. This picture over here is a picture of human papilloma virus or HPV. It is a double-stranded DNA virus. It is sexually transmitted, and persistent HPV infection over a long period of time can lead to cervical cancer development. But one should keep in mind that though HPV is necessary, it alone is not sufficient to cause cervical cancer. There are other various factors as well which are involved in the development of the cancer. Yes, you are hearing it correctly. A virus is even capable of causing cancer. One of the few cancers, rare cancers that occur due to viral infection, one such cancer is cervical cancer. So what are the other risk factors other than HPV infection, which is responsible for the causing of cancer? They are mainly genetic factors. And apart from that, there are various other cofactors like smoking, early age first birth and early age first intercourse, high parity. Parity means a huge number or large number of pregnancies that occur within a small time when your age is less. So that is high parity. Complete pregnancies. Having multiple sexual partners and also having low socioeconomic status. All these factors together can contribute to cause cervical cancer. So how does this HPV work? So what is the life cycle of this virus? The virus travels to the cervical region. It finds an abrasion or a cut in the cervical epithelium layer, in the topmost layer. It travels down until the basal membrane over here, that is the lowermost membrane, and attaches itself. It is undifferentiated, and as a result, the virus gets incorporated within that membrane. As the cells starts dividing, they along with the viral particles, they divide together and replication occur. Gradually, they move up, and through these stratified layers, they reach the topmost layer of the cornified epithelium. So this epithelium layer is sloughed off from time to time, just as you can see that dry skin or dead skin cells are sloughed off from the body every day. Similarly, mucous epithelium layer is also sloughed off. And along with that, viral particles are also sloughed off. They do not enter the bloodstream, and as a result, there is no viremia. But from skin to skin transmission, this virus can infect a body. How does the genome of HPV virus look like? HPV 16 is one of the most high-risk type of human papilloma virus that cause cervical cancer. This is the diagram which shows the genomic structure of HPV. You can see over here that there are various genes like E1, E2, E4, E5, E6, and E7. All these genes are early genes which are mainly responsible for functional proteins. So they code for functional proteins, the main machineries or the replication, transcription, all the important factors which are required are performed by these early genes. And these are the late genes, L1 and L2, which are mainly coding for structural proteins. They help in the formation of capsids of the virus so that it helps in the packaging of the virus. Now, E6 and E7, the early genes, are the major oncogenes that is responsible for the viral transcription. They are the main causal genes for cancer. E2 has a kind of suppression activity on E6 and E7. So E2 can control the transcription of virus within the human body or within the host. Now, in what state does this virus remain within the body? So it can remain in three different forms. The first being integrated. The virus finds different kinds of fragile sites in the human genome, and they integrate in those fragile sites together with the human genome and stay in that way. That is integrated. Second being episomal. The virus can stay in an extra chromosomal way. So the human genome is separate, the viral genome is separate in an extra chromosomal way. That is episomal in nature. And thirdly, there is a mixture of both integrated and episomal. That is, it can stay in between the two. That is known as concomitant in nature. Till now, I have described that how a virus is responsible for causing cancer. So you can see over here that there is an interaction of virus with the human body. The virus being the pathogen and human body being the host. So now I would like to highlight, this is a tale of two genomes where we are considering the host genome, that is the human genome, and the pathogen genome, that is HPV genome, both of which interact together very closely to cause cancer. What happens when the virus integrates with the host genome? As the virus integrates with the host genome, the E2 gene of the virus gets disrupted. And as I have mentioned earlier, E2 has some kind of suppression activity on the viral oncogenes E6 and E7. As E2 gets disrupted, E6 and E7 is highly expressed and they can cause cancer. How can they cause cancer? By interacting with various host proteins. So now let me come to that part. So we have all heard about P53, PRB. They are the main cell cycle regulatory checkpoint molecules that help in controlling the homeostasis of the body, like they maintain a balance, proper balance. As previously you have heard from Ankita, that there is a proper control that is being maintained from the cell cycle. So P53 and PRB are mainly responsible for maintaining the homeostasis. What will happen if P53 and PRB in the human body are disrupted? The viral particles, oncogenes E6 and E7 exactly do that thing. They functionally inactivate P53 and PRB, and as a result, the normal function of these genes, which is to keep the body in normal balance, in homeostasis, it is lost, and there is an uncontrollable growth of cells that occur, and as a result, cancer develops. E2 would have been present, it would have suppressed E6 and E7, and the homeostasis maybe would have been maintained. So this is how there is an interaction of the viral oncoproteins E6 and E7 with the host proteins P53 and PRB. Now, how can you say by just seeing or observing that you have cancerous cells at hand? So there are various characteristics which are important for cancer development. If you observe all these characteristics, you may be sure that you have come across a cancerous cell. They are mainly invasion, metastasis, genomic instability, resisting cell death, and one of the most important is avoiding immune destruction. The cells try and avoid the normal immunity of the body, which are cancerous. They avoid the immune system of the body and they uncontrollably divide. So these are all the characteristics which are called as the cancer hallmarks. And interestingly, it has been observed that the viral oncoproteins E6 and E7 can target each and every one of these interesting cancer hallmarks and cause cancer. So there is an integrate relationship of this virus with the host body. HPV is found to interact with both coding and non-coding human genes. Now we know that the human genome consists of only 2% of coding genes, while the rest 98% is junk DNA. But recent researches have shown that till now, whatever we knew as junk DNA is not junk at all, but rather though they are non-coding in nature, they play an important role in controlling or regulating the expression of this 2% of the coding genes. So it is equally or even more important than these 2%. HPV is found to interact with both coding and non-coding human genes and ultimately can cause cancer. So there are few genes which Ankita has already described, like oncogenes, tumor suppressor genes in the body. The expression of certain genes which are getting high in the body, they can cause cancer. They are known as oncogenes. And few of the genes whose expression is downregulated, they are tumor suppressor genes. Normally, they maintain a proper homeostasis and balance so that there is no problem within the human body. But when HPV comes, interacts with the human proteins, the levels of these genes get disrupted, and as a result, these genes they have an upregulated expression or they express much more and these genes they have a downregulated expression and they express much less in amount. So the normal homeostasis is disrupted and cancer occurs. Now coming to the most important thing, genetic factors in cervical cancer. How is the genetic makeup of a human body essential in developing cervical cancer? Now let's take an example. This over here is HLA. You can see this is a molecule that is human leukocyte antigen. It is present in the chromosome 6 of our body. It has 3 genes, HLA A, B, C. And what do these genes do? They mainly help in the antigen presentation. Antigen is any kind of foreign particles that can come from outside into our host, into our human body. And the immune system tries to recognize those antigen and kill it off or wash it off. So how will it identify? These are the molecules HLA-B, HLA-C, HLA-A. They try and present those antigen on the cell surface so that the immune system can come and kill it off. These genes are highly polymorphic in nature. So how does this polymorphism occur, or what is polymorphism? They occur when there is a particular substitution of or change in a nucleotide region in the genome sequence. That occurs due to single nucleotide polymorphism, and if it occurs in more than 1% of the entire population, it is regarded as polymorphism. And due to these subtle changes in the nucleotide sequences, different alleles are formed, and these different alleles are known as alternative forms of a gene. So a particular gene that contribute to a particular function may have variety of alternative forms. Like in this particular location, the capital A is present, and for the same location in another chromosome, small a is present. So these two are the alternative forms of a single gene which contribute to a single function but have different phenotypes. So they are alleles. So more the polymorphism, more are the number of alleles that are produced. And so these HLA genes, as they are highly polymorphic, different alleles are produced. So from our lab, it has been shown that if this particular allele is present in the human body, it is seen to be associated with cervical cancer risk. So the person has more tendency to develop cervical cancer. On the other hand, if this green allele is present inside the human body, it gives protection against cervical cancer. So you can say that a person who has this kind of allele in their genes, they are more prone to this kind of cancer. And if this allele is present within the genes, they are having or they can be protected against cervical cancer. This shows that variations in HLA gene can alter the capacity of antigen presentation and thus determine the risk of cervical cancer. Why do we need to study the genetic factors which is associated with cervical cancer? So let me say that cervical cancer is a late onset disease. It takes around 20 to 30 years to develop from the first HPV infection. It is a long time. So HPV infection occurs in normal cervix, and as a result, the cell morphology changes a bit, mild abnormalities develop, and so, but it may happen that the normal immunity of the human body comes into play, recognizes those infection and clears it off, so that it will again return to the normal cervix. But what will happen if there is persistent infection with human papillomavirus for a long time? So it will go on increasing and increasing, and it will progress and ultimately lead to precancerous lesion, which may be again of two types. It may be low grade or it may be high grade. If it is mild or low grade, it may again regress back. But if it is high grade, it will progress to cancer. From here, there is no going back. So this entire thing takes around 20 to 30 years to develop. So in between, in any stage, if an individual is tested, then she can be checked. Early detection if occurs, then she can be treated properly, and the cancer can be prevented. So that's why we are saying that this cancer is completely preventable. So early detection, or if exposure to causative factors is detected, it can be prevented. Now, genetic biomarkers are the most dependable markers over here, so on which you can completely depend and say that, okay, this samples, you can collect samples of individuals from these particular stages and test for different biomarkers present in it. So that particular biomarker will give you an idea in which stage that sample is present and how much progression has occurred. So for that reason, genetic biomarkers are important for the study of cervical cancer. Genomic factors, there are various epigenetic factors also which are important in cervical cancer. So we'll take the same example of HLA which we had discussed earlier. What happens in normal condition when HLA is present in high amount? There is antigen presentation because it is its function to present the antigen properly on the cell surface so that the immune system of our body can recognize that antigen, the foreign particle, fight it off and kill it off. So there is no cancer. But what happens during cervical cancer? There is HPV infection, and along with HPV, E7 oncogene comes. So under the influence of this E7, HLA expression gets downregulated. How does it get downregulated? There is kind of methylation in the promoter region of HLA. So from the promoter region is mainly responsible for starting of the transcription of HLA. So if the promoter region is somehow silenced, a silencing mark is placed on the promoter region, then there won't be proper expression of the gene. So the methylation mark causes a kind of silencing mark on the promoter region of HLA. And as a result, it is downregulated. Since it is downregulated, there is no or less antigen presentation on the cell surface. And as a result, our immune system can't find the antigen because there is no presentation. It can't fight it off, and so cancer progression occurs. So this is epigenetic because it is this kind of methylation mark is reversible in nature. It is not irreversible like the previous genomic factors which are present in the human body. There is a permanent change in the nucleotide in the genome sequence of a body, that is genetic factor, and here it is kind of reversible. Previous one was irreversible, and this is there is no kind of change in the genome sequence, and that's why it is epigenetic in nature. So how will you yourself identify or check for the symptoms of cervical cancer? This is for self-detection. If there is irregular vaginal bleeding or postmenopausal bleeding, bleeding after intercourse, vaginal discharge, pelvic pain, all of these signs and symptoms contribute to cervical cancer. So if anyone is experiencing these symptoms, she should report to the nearest clinic or go to the doctor immediately and get herself tested. So What is cervical cancer screening? Cervical cancer screening is just identifying that whether someone is having HPV infection or is developing or has a prone to develop cervical cancer. It can be done in two parts. The first being HPV testing or HPV typing. How? From the cervix portion, if you can collect the cells, it can be scraped off from the cervical portion. So those cell scrapes can be collected and DNA, RNA can be isolated from there, and particular PCR and real-time PCR based detection can be done to identify the presence of the virus inside the body. This is HPV testing. Or otherwise, pap smear test can be done. What is pap smear test? It helps in identifying the cellular morphological changes that occur. So it gives us an idea about an overview about the cell cytology. Cervical cancer thus can be prevented. So there are three main methods by which prevention can be done. Vaccines are available for this, which gives protection against any kind of HPV infection. So to protect yourself from HPV infection, one should get vaccinated against it. All of these vaccines are authorized by particular authorities. They are all approved by the authorities, and most of them are given at the age of puberty. Only Gardasil 9 over here can be given at a later stage as well. But India has no vaccination program or a screening program at national level. In recent times, you have heard about COVID vaccination. So there you have seen that how the government has aided in giving the vaccines. So since there is an aid given by the government, it is much easier. Otherwise, it would have been very much costly and normal mass would have not been able to get those vaccines. Same thing occurs over here in case of cervical cancer or against HPV vaccines. There is no such program, and as a result, it is very costly. The entire program takes about 8,000 rupees, which is not affordable by normal mass. But one can go and take the vaccines from the normal private hospitals. For that, you need to contact a doctor or private clinic nearest to you. Secondary prevention is screening for the presence of HPV 16 and 18, the one which I had discussed just now. So one should screen herself in every 1 or 2 years, because as I have told earlier, this is a huge process. It takes around decades to develop cancer. So in any point of time there is a proper persistent infection, you need to start your treatment. So for that, you should go and follow up your testings as well. Maybe every 2 years or every 1 year, you can go and get tested. Tertiary prevention is you should increase and spread awareness among your near and dear ones. You should understand how this disease pathogenesis occurs in terms of molecular factors. There should be more precise and effective therapies, affordable therapies. So for treatment, there are two types of treatments available. One being ablative therapy, where it is non-surgical, and another being excisional therapy, where it is surgical. So with these treatment methods, you can treat the viral infection or low-grade lesions as well. So I hope I have able to communicate to you all about the how cervical cancer occurs, how it can be treated, prevented. So early detection is very much important, and hopefully you all will use your the resources which are available with us properly. So in NIBMG, there is a lab which works under Professor Shormila Sengupta's guidance on cervical cancer. We screen samples in Kolani cohort, and there are also different camps which are being organized. Various genomic studies are done in our lab, and so from there we can get insights into the cervical cancer pathogenesis. I would like to thank Professor Arindam Moitro, Dr. Alpana Dave, and Science Setu for giving us this platform, DBT. And thank you all for listening. Hope you have a wonderful life ahead. Thank you.
Thank you, Abarna. There are a couple of questions for you guys. So, do you have access to YouTube, or should I?
Yes, we can see the question. We can see the question. The first question is from Ritam Chatterjee, who has asked about. Okay, let me just go there. Okay, so there is one question from Ritam Chatterjee who has asked about. I'm reading out his question. Oral cancers are often preceded by pre-cancers like leukoplegia. Have you seen the genetic expressional status for those? Very interesting question and very valuable question indeed. During my full presentation, I have not talked about precancerous lesion, but yes, precancerous lesions are very important. Like when I have mentioned about the early detection methods, and I have mentioned that there could be some white patches. So those white patches are, we call it as precancerous lesions. They can be of different type. Leukoplegia is one of them. And what happens is prolonged tobacco exposure sometimes gives rise to those lesions. Those lesions can be benign, but they can progress to cancer. Approximately 4% of the patients with leukoplegia progress to cancer. And there is a recent ongoing study in our institutions by Professor Partham Pradeep Majumdar and Dr. Nidhan Biswas, who are studying the genetic variants or mutations common between the cancers and the precancerous lesions. So if it is seen that for patients who have cancer in one cheek and precancerous lesions nearby, there is approximately 65 to 67% of overlapping mutations carried by both the tissues. And yes, this study is still ongoing, and we are looking into the genetic determinants of precancerous lesions and trying to identify which ones could progress to cancer in late stage and which ones are simply benign. So I hope I have answered your question. Thank you, Ritam. There is another question by Abhishek Roy. I'm reading out his question. Is HLA allele mutagenesis has any relation with PD-L1 expression which leads to evasion of immune system? So I think this question is for Abarna, and Abarna will answer this.
Hi. So yes, there is. What was the question? Yes, HLA mutagenesis has any relation with PD-L1 expression. Yes, it is correct that HLA has a kind of relation with PD-L1 expression. In our lab, our seniors have worked with this as well. The main purpose of it is antigen presentation. So since it is down regulated, what happens, they are not being able to actually being recognized by PD-L1 or PD1 genes, which are usually responsible for T cell immune responses. So as a result, it cannot act properly. And so what happens, the cancer progression takes place, and the immune system is also disrupted over here because of that. And so I can say this much in because it requires a lot of details, and I would like to say this much only, that yes, there is a kind of relation, as both of them is responsible for normal immune defense in our body. And So, is there any data available for Indian cohorts related to cervical cancer like TCGA, CESC? Okay, till now there is no Indian cohort data which is available like TCGA, CESC dataset. TCGA, CESC is mainly, as you know, it is for the Western countries or for that. In Indian cohort, there is no such data on cervical cancer. Though we over here in our lab are working with various samples. We have a huge number of samples which for which we have tested and sequenced, and we are collecting the data. Maybe in future we can develop some kind of database for everyone in India. So I don't think that's it. Okay, there is another question. Pre-cancerous stage of cervix. Yes, pre-cancerous stage of cervix, it is known as L-cell and H-cell, that is low-grade cervical intraepithelial lesions and high-grade cervical squamous intraepithelial lesions. These are the two categories which are pre-cancerous in nature. So, as I mentioned earlier, that this take a huge time to develop. If it can be intervened in between these stages, it can be treated, and maybe it can be prevented. Okay, so there is another question. What are the non-coding genetic factors for cervical cancer? Okay, so I was kind of hesitant in saying this because it is a bit difficult. Okay, but I'll try and explain in easier language so that you all can understand. So non-coding genes are mainly responsible for regulating coding genes, right? For regulating the expression status of different coding genes. So in cervical cancer, there are various reports of non-coding genes like hot air. I hope you have heard about hot air. It is one of the most important non-coding genes which are responsible for cancer. So they mainly try and act on various coding genes and manipulate their expression. So epigenetically also, genetically also, what happens? Hot air can come and interact with various E7 oncoproteins, with the E7 oncoprotein. And as a result, what happens? The normal function of the genes like chromatin remodeling complexes, which are mainly responsible for maintaining the compactness of chromosome, they are disturbed. So when the chromosome or the chromatin modeling complex stays in normal condition, there is a particular type of gene expression, but when hot air comes into action along with HPV or E7, there is a kind of silencing mark or epigenetic mark that is created in the promoter region, and as a result, the chromatin remodeling compaction is lost and it is a bit of different. And as a result, the downstream coding genes expression patterns are changed. So those coding genes may be responsible for the progression of cancer. They may be oncogenes whose expression is highly expressed. They may be highly expressed, or they may be tumor suppressor genes also. So for the example I'm giving right now, if they are silencing marks, then it will be for the tumor suppressor genes. So if it is silenced, the tumor suppressor genes won't work. So I have given only an example of one non-coding RNA. There are numerous other non-coding RNAs which work like recently we have found something like antisense transcripts of a particular coding gene can act against that coding gene. That antisense is non-coding, but it can act against the coding gene and disturb the function of the coding gene. So proper function of the coding gene is lost. So this is how non-coding genes can interfere and can cause cancer as well. Oh, have you found any novel genomic alterations related to cervical cancer in context to Indian cohort? Yes, we have found many novel genomic alterations in context to cervical cancer in Indian cohort as well, both genome coding and non-coding. So studies are ongoing now. So it will be beneficial if you can visit our webpage also. There are various papers available on that, and there are numerous genes which are responsible, which are actually novel in nature, and only in Indian cohort itself, we are actually studying on the Indian cohort, so we can say that it is novel in Indian cohort only. There are various immune genes which are absolutely exclusively present in this Indian cohort and not in the TCGA dataset also, which is exclusive for our dataset. Okay, I guess there is no other question. So thank you everyone for listening. Hope you all have got an idea about oral and cervical cancer now. Thank you.
Thank you to both the speakers for their fantastic talks, and I'm sure all the attendees have gained something out of this and would now be able to prevent or at least create awareness about these two cancers. So thank you everyone for joining in. Take care.